everyone, and welcome to my channel. This is the ninth episode of the Meet the Pro series, a series where I interview professionals in the field of biology and genetics to help high school students interested in the subject. In today's episode, I will be interviewing Dr. Shiv Pillai. He's the director of the Autoimmune Center of Excellence at Mass General, a core investigator at the Ragon Institute of Mass General, MIT, and Harvard, and a director of the Harvard Immunology PhD program. I am delighted to have him with me here today. Thank you for being here, Dr. Pillai.
I'm happy to be here, Rishi.
So, could you take a few minutes to describe how you first became interested in biology to eventually getting to where you are today?
I guess when I was in high school is when I got excited about biology, and somehow I think when I was about 13 or 14 years old, there was a version of a book at home. It was called Microbe Hunters. It was actually an abridged version of the book, I think. It was one of those condensed books from the Reader's Digest. I read the real book later. And it was the story about Pasteur and Koch and the discovery that diseases were caused by pathogens and, about that era. And it is that era that changed medicine from being a field where people believed that diseases were caused by some imbalance of humors. The people thought there was this imbalance between blood, bile, black bile, and phlegm. And that's why there were therapies like, application of leeches, bleeding people basically, giving them emetics so they would vomit, so to get rid of the phlegm. And they were thought this balance had to be corrected. That was the view of disease for 2000 years. It wasn't very different in this is the Western system of medicine, which came to most of us through the Greeks, via the Arabs, then to Europe, and then to America. And this was the view till about 1870 or so. In the 1850s, people started to doubt it. So this book was one of those great books explaining how this change occurred and how people hunted for microbes. And I was just fascinated, and I thought, wow, this is what I want to do. And then I thought, to do that, I have to go to medical school. I didn't know any better. I mean, I could have just become a scientist maybe, but that wasn't even when I grew up in India, you were either going to become a doctor or an engineer. And if you're good at math, you're supposed to become an engineer. I was good at math, but I really liked this. So I chose to go to medical school. And when I went to medical school, I was fortunate to go to a medical school where there was some decent science being done. And people who interviewed me were interested that I was interested in research. So I ended up doing research as a medical student. I ended up doing a PhD. And that is how it all started, from that interest, that early interest in, this fascinating story about how we learned about disease.
How did you come to choose an academic career rather than one in the industry?
Oh, I see. I mean, that wasn't a difficult choice at all. So, first of all, let me tell you how the world is structured, even in America right now. Almost every important discovery is made in academia. Some of those discoveries get taken by academics into biotech companies, which then take it all the way that you can create a product that might get into a patient. Typically, the way it works today, the pharma companies then buy the biotech company. So then they can do clinical trials with that drug because they have the money to do it. And that's how we get products. So if you think about where is discovery happening, that is in the academic world for the most part, right? And the fundamental discoveries are what then drives application later on. So if you go back and think about, as some of your viewers might know, we now have cures for cancer. Where did those cures come from? The cures came from understanding how the immune system works. And those are fundamental questions, and then you could apply them. It kind of works both ways. If I go back and think about Louis Pasteur, Pasteur was trying to solve the problems of the wine industry when he discovered fermentation. So it came from a different angle, but the real thing is that whatever you start with, you want to do good science. And when you're doing good science, then you learn something fundamental from it, and that might be applied much more widely.
I also saw that you were very interested in theater and poetry, and like I watched some of your TikToks about COVID. Could you speak a little bit about how these passions influence your teaching style, if at all?
I mean, they do influence my teaching style for no doubt about it. So, when I was young, I was heavily into theater. I was part of we part of a group that started one of the early English language theater groups in the city of Calcutta,
Hmm.
and that theater group is still going strong now. About it's more than 55 years later. It's still it's produced a lot of famous movie stars too in India,
Hmm.
but this was all in English. So I was into theater. Did theater in medical school. That was a passion. That's an interest,
Yeah.
and started from high school. And when I started teaching, then of course I decided I wasn't going to choose a life in theater. That was a theoretical possibility, and
Yeah.
I was really interested in doing science related to disease. So I ended up doing this PhD and then coming out to the US to do a postdoc. All of that happened. And when I started teaching at Harvard, when I was teaching the undergrads, I think the first year I was teaching, I realized they needed some entertainment. So
Okay.
then, I mean, this is their undergrad, right? I mean,
Yeah.
they love. I used to tell stories. They like stories.
Hmm.
All right, let's do some summary in the middle of the course. So I did a thing called the lymphocyte rap. I wrote up something on a flight to Greece. I
Okay.
had to go for some meeting in Greece. So I wrote a
Hmm.
rhyme on that, and I came back and performed it for
Yeah.
the class. Then, being from theater, I cannot think of reading something. I have to memorize it and perform it, right? That's the way I do it.
Okay.
So that's what I did in class. And
Yeah.
I did that 1 year. I was teaching the undergrads, and then a few years later, I had like four rhymes. It grown a bit. People like
Hmm.
my
Hmm.
doing it. Then I had a fairly outspoken. When I say outspoken, I mean someone who didn't ask me first. Basically, I had a teaching fellow, one of our PhD
Uh-huh.
students,
Okay.
who stood up on the first day of class at Harvard and said, "The professor will have a rhyme at the end of every lecture." And I
Okay.
had 20 lectures and about
Yeah.
four rhymes.
Okay.
So that year, I worked very hard.
Yeah.
For every topic, I have a rhyme, and I have to memorize them.
Yeah.
And I have them all pretty much memorized, and I just do them at the end of class. It's fun for me.
Yeah. Yeah.
I tell my students I'm a frustrated thespian in search of a captive audience.
Okay.
I enjoy it.
Yeah. I actually I watched your lymphocyte rap on YouTube.
And
So
by
yeah.
the way, I mean I have never put anything on YouTube. My
Oh,
students will
yeah, your
quietly
student.
film it and put it on YouTube, and
Yeah.
I say, okay, who gives a damn? I mean,
Yeah.
do what you want. I don't mind. Yeah.
Okay. Could you talk a little bit about your work at the Ragon Institute?
Yeah, so my lab has been studying the immune system for a long time.
Hmm.
Started from the days when I started out as an assistant professor more than 35 years ago, I guess. And the research we do has focused on trying to understand some fundamental principles of how the immune system works and how it relates to disease, in the hope that we will learn things that will help us, sometimes treat diseases as well. It's never been aimed at, let me find a cure for this disease.
Yeah.
It's always been aimed at look at the process.
Hmm.
So we were fortunate that a long time ago we figured out we're trying to understand how immune cells make sure that they have only one receptor, right? You have
Yeah.
two
Yeah.
genes. For every gene, you have one copy from mom and one from dad.
Yeah.
But our receptors, which are made in a very intricate way, the immune receptors are unique and amazingly diverse. And when we make these receptors, somehow we have to make sure we make it only from either from mom or from dad, not from both genes. We have
Yes.
two receptors in an immune cell,
Hmm.
then it loses specificity, right? It becomes
Yeah.
specific
Yeah.
for more
Yeah.
than one thing.
Yeah.
So becomes really important. So I try to work on that question earlier on, and then we did get an answer. But in that answer, we learned about a pathway, and there was an enzyme in that pathway.
Hmm.
And we recognized that maybe this enzyme would be useful in treating patients since it was involved in how the B cell signals, a lymphocyte, one of the immune cell signals, and multiplies.
Yeah.
So maybe in cancers of the immune system or in autoimmunity,
Hmm.
when you want to dampen things, maybe, this could be a target.
Yeah.
Most drugs before the 1990s, almost all drugs, were basically inhibitors of enzymes. They were small molecules
Yeah.
that could bind to. So enzymes always have a something called a hydrophobic cleft. It's a patch.
Mm-hmm.
into which the drug can go and sit, and so it can
Yeah.
bind tightly. So most drugs are these kind of drugs that go in. Nowadays we have new drugs we call biologics, right? They're different. So this enzyme was one of the first enzymes that was shown to be key to activate a type of immune cell.
Mm-hmm.
So people made inhibitors to it. We didn't make them.
Yeah.
We described the enzyme and showed
Yeah.
what it
Yeah.
does, how does it work, all that stuff. And then there are about 9 or 10 companies which make an inhibitor to this enzyme, which is used for it's the treatment of choice for patients with a particular type of leukemia, which occurs in older adults,
Mm-hmm.
chronic lymphocytic leukemia. So that was one approach that we did a long time ago. Then since then, we've been trying to ask you what is driving an autoimmune disease, and then how does some therapies work, and how do they work? What do they do? How does the immune system get involved in it? So that's one of the things we studied. Then in COVID, we were intrigued by the fact that in severe COVID, you lost one type of immune cell called one type of T cell. But I think what we figured out that why we lost that cell is that this is a sort of a stereotypic response in the immune system. We have a very severe infection.
Mm-hmm.
You want to make sure that the immune system doesn't get too active and kill yourself, right?
Yeah.
So if your immune system is trying to wipe out a virus, and
Mm-hmm.
the virus infected 3/4 of your liver, let's say, right? Then if it wiped out all those infected cells, then you will have no liver left and you would
Yeah.
die.
Yeah.
So the immune system evolved a mechanism to maybe make those cells quiet and do it less efficiently.
Mm-hmm.
Then it needed an alternative to get rid of the virus.
Yeah.
And I think that's why we think we lose these types of other types of immune cells which cause a Darwinian structure to develop called a germinal center, because you don't need that yet. And we
Yeah.
use those cells in a different way to try to
Okay.
clean out the virus. So that's one of the things we worked out the mechanisms and we study.
Mm-hmm.
And then we try to ask, what types of cells are causing disease, what are not causing disease. One of the students in my lab is trying to figure out why you get anaphylaxis. You get allergies, but why do you get anaphylaxis? What is so different about what's going on there? And
Mm-hmm.
that's also turned out to be quite interesting. So there's many different angles
Yeah. Yeah.
that we work on. It's largely related to how we basically think, and we are true believers in this.
Yeah.
It's reality. It's not just belief, by the way. It's
Yeah.
that
Yeah.
almost all diseases are caused by some imbalance of the immune system.
Mm-hmm.
You might have a genetic basis for it
Yeah.
in some people. You might have an environmental trigger for it.
Mm-hmm.
But fundamentally, it's when the balance in the immune system gets lost.
Yeah.
That's when you get disease.
You mentioned earlier about how um there's the possibility, or like why we didn't have two antibody genes, even though we have the maternal and paternal, and why there's only one that's expressed. Um, like for normal traits, things like hair color, skin, eye color, things like that. Those traits, if those are expressed based on like dominant and recessive, does that not apply to these antibodies?
Yeah. So when you think about dominant and recessive, what you really mean is that if one of the genes, say mom's dad,
Mm-hmm.
is going to be more clearly expressed
Yeah.
over dad's gene, let's say that, right?
Mhm.
So then we say it's dominant. It's
Yeah.
got maybe a change which just the product of that gene with the change, that allele with the change, is going to be the product that you will see. So
Yeah.
I'll explain it that when you think about this, you don't think about this as in normal traits. Normally, you would think about this in the context of disease. So if I have two genes, and then one gene got a mutation that makes it super active, one allele. So let's say mom's gene got a
Mhm.
mutation, makes it super active.
Yeah.
Okay. So now I have a problem. This super active guy may do something that does damage to me. Dad's
Yeah.
gene is not causing it. So in that case, that's a dominant mutation causing a
Yeah.
disease.
Yeah. Yeah.
In another case, so typically for most situations outside of the sex chromosomes, for most
Mhm. Yeah.
genes, both copies
Mhm.
of the gene are contributing, right? If I have a
Yeah.
level of a protein, about half is coming from one, about half is coming from the other, roughly. That's generally the
Yeah.
case, right? There may be small difference in expression between maternal and paternal,
Mm-hmm.
but that's it. In the case of a recessive disease, both copies, either one gene got lost from mom, let's say, and
Okay.
dad's gene got mutated, or both genes got mutated.
Yeah.
So that's a recessive trait, right? So again, we would think of that more when you have a change or a trait, right? For
Mm-hmm.
most things, we're using both copies. And we're using, the most of our function is coming from both copies. Sometimes we can lose one copy completely and still the other copy can do enough for us and it doesn't matter, right?
Yeah.
But in this case, we are saying, no, you cannot allow each immune cell, cannot allow both mom's and dad's genes to be functional at the same time.
Okay.
If it does one first, it must shut off the other. So we call that allelic exclusion, where there's a need to make sure that every cell
Yeah.
will not express both products.
Mm-hmm.
And it's not like the gene is defective, right? It's just that
Yeah.
one is being used and the other is not. And how does that happen? That's what we were interested in figuring out. That was allelic exclusion. When I started on that work as a postdoc, I thought this will be the answer to autoimmunity. That's why I should do it, right? That was
Yeah.
the way I thought.
Yeah.
That if you lose specificity, you lose. So it's important, of course. It's important for the development of B cells and T cells, but that's not. It's a different story.
Yeah. So I mean, this next question might also be related to this, but you coined this term called surrogate light chains. What does that mean? And was that related to this allelic
Yeah.
thing?
So basically, I mean, I'll try to simplify this. An antibody has two chains, a heavy chain and a light chain.
Yeah.
But we always make the heavy chain first
Mm-hmm.
because it's a complicated process. We make the heavy chain first, and every time we make the heavy chain in a certain cell,
Mm-hmm.
why do we make it first and say that now this heavy chain protein can assemble with any different light chain that you make? We're trying to create diversity, right? In the immune system. So now we have the heavy chain and say now any light chain can combine with this. If you did them both together at the same time, then you're fixed. You're not using the diversity of one to create a diversity.
Yeah.
So the process that we found was when you make the heavy chain first, the cell wants to know you've made a heavy chain. And if you made the heavy chain, it says, I need to know and I need to shut off the process that will occur on the other chromosome. Let's say this was dad's chromosome. I want to shut off mom's chromosome, right? That's what it is.
Yeah.
In order to do that, the cell, how it recognizes that it's made the heavy chain is it has a pre-existing chain that looks like a light chain. It's not a regular light chain for diverse immune recognition. We call it a surrogate light chain. We discovered this protein, so we call it a surrogate light chain. And its job is to basically identify that you did the good job and made a good heavy chain protein. That's all. And then it shuts off the chance of making a second antibody gene. Really.
So these surrogate light chains are like they're what ultimately maintains this specificity within the antibodies.
Yeah, they're only during the develop a B cell. You can think of a B cell is born, it's a baby, then it's a teenager, then it's an adult. So it's in that early stage when it's trying to make its receptors. It's only then that it needs the surrogate light chain. So it comes on, does its job. When the B cell has become mature, then it's gone. So now the B cell doesn't need it anymore. That's how it is.
Okay. Are there any common difficulties you encounter when trying to cure immune system diseases? Or I mean, I guess as you said, that's not
No, I mean, so the reality is this. We can actually cure many cancers, not all, about 20-25%. When I say we, I mean the whole field, right? 20-25% of cancers can be cured. There's some clever ways to do it. People are thinking of very clever ways to cure autoimmunity. There was a paper that just come out which may do it for some autoimmune diseases. But in general, we don't believe we have yet learned how to do it. And if we can learn how to do those things, for which I think we need more knowledge to be able to do it right, then we may be able to cure many common diseases. Like, we cannot cure most common disease like hypertension, asthma. You can't cure. You can just treat them, right? The question is, can we cure them?
Okay.
And so that's why we feel that deeper knowledge of this will allow us to understand it so well that you can now think of a cure.
And finally, I want to end with a sort of fun question. If you could choose any extinct species to bring back, which one would you bring back and why? And you cannot say dinosaurs.
I cannot say dinosaurs, right? An extinct species.
Yeah.
And
Or they
I'm
can even be like very endangered, but endangered
Yeah,
to.
I'm thinking of a couple of endangered species
Mhm.
which will probably vanish maybe in your lifetime, may not be in mine.
Yeah.
And one is called the cheetah. Have you heard of the cheetah? You heard of cheetahs, right?
Yeah.
Cheetahs may vanish, and they will not vanish because humans are getting rid of them. They
Hmm.
may vanish because they have a big gap in their immune systems. And the other is an animal called the cotton top tamarin, which is in South America.
Okay.
The cotton top tamarin also may vanish, again, not because of being hunted, maybe some of that happens
Yeah.
too, but because it's got this similar big deletion in its immune genes.
Why do they? Why do the cheetah and this and the cotton the
So
other animal?
somewhere along the way, if you're heavily outbred, you may never face these issues, right?
Okay.
When a species has become very inbred,
Hmm.
and if by accident has a founder deletion mutation where you
Hmm.
lost a big chunk of your chromosome,
Hmm.
right? And then for some reason, because there's brother sister marriages or matings can happen in nature, right?
Hmm.
If this kind of thing gets fixed in the population,
Okay.
then you have this problem that you have a population with a very tight, very not diverse genome
Hmm.
in which there's a big deletion in one aspect of something or the other, right? In the
Yeah.
case of the immune system, you can survive, but because you lost that type of thing, so the cotton top tamarin, for instance, gets a lot of lymphomas, gets
Hmm.
lots of viral infections, and then
Yeah.
it dies of these.
Okay.
So your lifespan is already reduced.
Hmm.
Now your reproducible age is not as good as it should be
Okay.
when you have all of these things.
Yeah.
So the population declines. So in
Yeah.
these are things that obviously if we could fix these guys, I mean, it's
Yeah.
not like I particularly love cotton top tamarins,
Yeah.
but I just think of in nature,
Yeah.
this is a means of loss. It is
Hmm.
not, we don't have to think about asteroids hitting the earth and everything else. These kind of losses can happen. As populations narrow, they can accumulate defects and sometimes you can lose them.
Okay. Well, I think that's all we have time for today. Thank you so much, Dr. Pillai.
You're very welcome, Rishi.
Thank you.